So today we are having another chapter, that is the enzyme. Chapter 2, that is the name is enzyme. Before that, chapter 1 we have discussed about that was the biomolecules, and which we have discussed about the four biomolecules, that was nucleic acid, protein, lipids and carbohydrates. Now we are having enzyme. First of all, what is mean by enzyme? Enzyme means biocatalyst. Biocatalyst. What are biocatalysts? Biocatalysts are those that speed up the chemical reaction. That speed up the chemical reaction, that are called the enzymes. Biocatalysts that speed up the chemical reactions, that called the enzymes. We have different types of enzymes. First of all, these are the catalysts. Those catalysts are those who accelerate chemical reactions, but themselves but themselves but themselves remain unchanged before during and after the reaction. What does it mean? That catalysts are those who accelerate chemical reaction. Accelerate means who speed up the chemical reactions, but themselves will remain unchanged. They will not change themselves during, or we can say after the chemical reaction. They will remain unchanged. They will be having same structure and same function, but they will be only catalyzing that product. So these are the catalysts who accelerate the chemical reaction, but themselves remain unchanged during and after the reaction. But we are having another that change themselves after the chemical reaction. After the chemical reactions, called activator. Activator are those who change themselves after the reaction. Enzyme do not change themselves, but the activator change themselves after the chemical reaction. Or we can say, or activator are those who involve in that reaction, in that reaction. Basically, enzyme were not involved in enzyme, were only catalyzing that product, but the activator will be involved in that. It will change its, we can say, structure after the chemical reaction. After that, we are having some characteristics of enzyme. We are having some characteristics. First of all, before characteristics, I will discuss some point that we can see it was considered as a, which means enzyme was considered as that all enzymes are proteins. It was considered as that all enzymes are proteins. In 1980, there was a scientist called Thomas Catch and Sidney Altman. These both were the scientists. Basically, they discovered that some molecules of ribonucleic acid when they work as enzyme, when they or we can say work or function as enzyme, so that molecules are called ribozyme. In 1980, there was a scientist called Thomas Ketch and Sidney Altman. They discovered there is some molecules of ribonucleic acid. As we know, what is ribonucleic? It is a hereditary material. So ribonucleic acid, when they work as an enzyme, they are called ribozyme. They are called a ribozyme. Basically, what are ribozyme? These are the coenzymes. Now, what are coenzymes? Coenzymes are those who work with the enzyme, or we can say who help the enzyme. So, basically, ribozymes are the non-proteinous. These are the non-proteinous. That's why these are the coenzymes. So, after that we were discussing about the characteristic of the enzyme. So we are having some characteristic that the number one is the most of the enzymes are proteinous in nature, some of them are non-proteinous, as is ribozyme. In nature, some enzymes are not proteinous. After that, some enzymes are pure proteinous. They name as a simple enzyme. After that, we have another term that some enzymes have non-proteinous and proteinous part both, so they are called the conjugated enzyme. As if you remember that in previous video I discussed about the conjugated molecules in the biomolecules, right? Conjugated molecules were like, if the lipid will be added with the protein, it will make lipoprotein. So as here, that when proteinous and non-proteinous part will work, or we can say an enzyme having these both parts, they are called the conjugated enzyme. Conjugated, which means conjugate, to combine. So that is conjugated molecule. Now in conjugated enzyme, we are having two parts, right? Number one is proteinous and another is non-proteinous. Proteinous and non-proteinous, right? Now proteinous part is called the apoenzyme. Proteinous part called the apoenzyme, and non-proteinous part is called as a prosthetic group. Now in non-proteinous part, we are having another two categories in prosthetic group. Number one, it is cofactor, and number two is the coenzyme. As here I have discussed about the coenzyme, so here you will clear about what is coenzyme, right? Basically, cofactors, these are the inorganic in nature. These are inorganic, and coenzymes are the organic. These are the organic. Now inorganic, these are also known as the activator. These are also known as the activator. What is mean by activator? That activate that enzyme, that activate the enzyme that called the activator, right? Now what is mean by coenzyme? Basically, coenzymes are the organic. Like, we can talk about the NAD, FAD. These are the coenzymes, right? So these are the organic. Now if we talk about the inorganic, like inorganics are the salts, or we can say metallic ions. These are the inorganic. For example, we are having magnesium, NaCl. Basically these are the inorganic nature. Basically these activators. These are activators, or we can say cofactors, that will activate the enzyme. These will be activating that enzyme, so that are called the inorganic in nature. After that, we are having another point that some enzymes act within the cell. They are called as endoenzyme or endozyme. After that, some enzymes act act outside of the cell. They call the exoenzyme. So if we talk about the example of endoenzyme, that is a adenylate cyclase. This is an endoenzyme that is work in within the cell or in the cell. So this will speed up the reaction in the cell, inside the cell, or we can see. So the enzyme that work within the cell called the endoenzyme, that work outside of the cell, that is called the exoenzyme. After that, size of the enzyme is greater than the substrate. Greater than the substrate. What is mean by substrate? Now substrate is that, for example, one we can say product, we can say particle, that we want to make the product of that one, that is called substrate. Like we are eating food, right? So when we eat food, the enzyme will be attached with that, so that is called substrate, which means the size of the enzyme is larger than substrate. Which means if the substrate will be larger than the enzyme, so how substrate will be attached to the enzyme? Here we have to attach the substrate to the enzyme, so enzyme should be larger than the substrate, right? That's why substrate will be attached to the enzyme. After that, we are having another point that enzymes are specific in nature. Enzymes are specific in nature. How in nature or the function, how every food, or we can say every molecule, have its own enzyme. For example, protein. For the digestion of protein, we are having a particular enzyme called the protease. For the digestion of the lipid, we are having particular enzyme, that is the lipase. For the we can say digestion of the carbohydrate, we are having another enzyme that called amylase. So these are the particular enzymes that will work only in particular nature, because in a specific nature. Enzymes are specific in nature, like every molecule or every substance have its own enzyme that will only work on that molecules nature, not on other nature. For example, we have to do the digestion of the protein. Lipase will not work, only protease will work, only and only protease will work. That's why every, we can say, for the protein, for the digestion of lipid, for the digestion of fats, for the digestion of carbohydrate, we are having different enzymes. That's why I call the enzymes are specific in nature and in function. For example, protease cannot digest lipid, it will only digest protein, so that's why it's called as a specific nature. After that, enzyme are active at a specific temperature or pH, which means enzyme will only work at their specific pH. For example, for the digestion of protein, they are having pH, for example, 6.8, that's slightly acidic, right? So the enzyme protease will only work on that pH. Enzyme are active only at a specific pH. If any enzyme having its own pH, for example 7, that will only work on that pH. That's what's called the specific pH or temperature. After that we have another point, that if conjugated enzyme, if conjugated enzyme work as a simple enzyme, that is called as a holozyme. What was a simple enzyme? I have discussed here. Simple enzyme are those enzymes here. Simple enzymes are those enzymes, those are pure protein enzymes, pure proteinous in nature, that are called simple enzymes. We can say conjugated energy. What is conjugated enzyme? That are the proteinous, non-proteinous both. So when conjugated enzyme will work as a pure proteinous nature, that will called as a holozyme. So if we take it the example, for example, we are having pepsinogen. We have a pepsinogen, that is inactive enzyme. When we add activator with this activator, before I discuss what is activator, that are the inorganic in nature. So HCl is the salt, right? That is activator, that will activate this pepsinogen. This will be called as a conjugated enzyme. They will provide as a conjugated enzyme. So when activator will, we can say, work on the pepsinogen, it will give the something that called the pepsin, which means now when HCl work on the pepsinogen, it will come in its active form called the pepsin. It will come in its active form called the pepsin. So this pepsin is known as a holozyme. This pepsin is known as a holozyme, right? After that, we are having one model called the key lock model. Key and lock model. Basically, this is a model of enzyme, right? So I'm discussing here. So basically, what is meant by key and lock model? Here, lock plus key model. This was proposed by Fischer. He was a scientist who proposed this key and lock model. Now, this we can say this model was improved by D.D. Wood and Fielder. Now, what was basic statement of this specific enzyme? React with a specific substrate. As I discussed, what is substrate? So a specific enzyme, which means, for example, this is an enzyme and the specific substrate, the substrate which is of this enzyme, that will only attach with this. If there will be substrate like this cannot be attached, because you can see the size of that enzyme. So what is meant by key and lock? Basically, this is a lock. If you will be putting right key in this, so that will be working as an enzyme. It will be putting wrong key in it, so that will be obviously not work. So this is work as a key. That's what is known as a lock and key model. So a specific enzyme react with a specific substrate. After that, another point, that is the active side is rigid. Active side is rigid. Now, what is mean by active side? Here, for example, this is enzyme. Basically, this side is known as a catalyst side. And this side is known as a binding side. And these both, when we call these both as an active side, this binding side and this catalyst side, these both are known as an active side. So here saying that active side is rigid means fixed. So this active side is fixed. If the substrate will be attached of this size, so it can attach this, but it is a rigid, it is fixed. If another size, for example this size is attached with this cannot be attached because it's rigid, it cannot change its form according to the substrate. So that was a key and lock, we can say, model. After that is another model called the induced fit model. Fit model, there is a point in this model that active side is not rigid. Active side is flexible. It is flexible. How? Like this is enzyme. If the size is this, so it can attach. How it will be attached? It can attached by its size and flexible itself. So the active side is flexible. It was proposed by induced fit model.